Thank you, Herman. Well, welcome everybody to our final installment in the Live Longer, Live Better lecture series. Today we have Heinrich Jasper with us. He's here to deliver the Orkland Lecture. So the Orkland Lecture is named and endowed in memory of Richard Orkland, a Ph.D., a pioneering researcher in the field of glial neurobiology. Glial cells make up the connective tissue of the nervous system and play a critical role in regulating the neural microenvironment. Orkland earned his doctoral degree at the University of Utah in 1961, served on the faculties of the University of Utah, UCLA, University of Pennsylvania, before becoming director of the Institute of Neurobiology at the University of Puerto Rico. He spent several summers working at the biolab. His friends remember him as an ambassador for glial research and for his, quote, rare blend of talent, humor, and joie de vivre. So Dr. Jasper, as I say, is with us today. He obtained his diploma in biochemistry in 1999 from the University of Tübingen in Germany. He received his Ph.D. in 2002 from the University of Heidelberg and the European Molecular Biology Laboratory, where he studied transcriptional regulation of developmental processes in Drosophila. He was appointed research assistant professor at the University of Rochester Medical Center in 2003 and became an assistant professor in the Department of Biology there in 2005, where he's promoted to associate professor with tenure in 2010. I know him from when he came out to the Buck in July of 2012, where he served as its chief scientific officer from 2014 to 2017. Then, about a little over a year ago, he was appointed staff scientist at Genentech. I like to say they probably made him an offer he could not refuse. Henry's work is focused on stress and inflammatory signaling pathways that influence tissue homeostasis and longevity. His lab has investigated the regulation of somatic stem cells and the many intrinsic, local, and systemic cues that control how the stem cells behave, both in homeostasis and in disease conditions. A particular focus has been understanding how age-associated chronic inflammation perturbs regeneration in barrier epithelia. His lab has also explored mechanisms regulating photoreceptor cell death and survival in the injured retina, and have identified strategies for immune modulation that increase the efficacy of cell replacement therapies in this tissue. So he's very interested in exploring the translational strategies based on his work. And so he's with us today. Welcome, Dr. Jasper.
Thank you so much. Oh, this is on, okay. I guess you can hear me. Thank you very much for inviting me. It's a pleasure, it's an honor as well. I was here last year and I had really a lot of fun, especially in this setting, which is so beautiful. What I want to do is give a little bit of an overview of what we've been working on over the last almost 10 years. And work that was primarily done at the Buck Institute. Let me see if this works. Yeah. Which is up in California, but I have a small lab in Germany as well, and this was actually up until last year, and still have these labs, but I'm slowly transitioning, as Eric said, into Genentech, and we're ramping up the work there, but I don't have any data to present yet. So I'll talk mostly about work that was done at the Buck Institute, where we've really tried to explore how aging influences stem cell function in somatic tissues, how ultimately aging impacts the regenerative capacity of many of our tissues. And we were trying to identify potential strategies for intervention in which we could improve regenerative capacity in these tissues. And we use primarily the fruit fly as a model system for this, but have recently also moved more into vertebrate systems. And I'll give you some examples for how the work in fruit flies really can inform our understanding of stem cell function in the vertebrate system. So fundamentally, what we're interested in is in stem cells that sit in most of our tissues, which are ultimately cells that have a multipotent capacity They are maintained usually by what is called a tissue niche, a microenvironment of cells and extracellular matrix that maintains this multipotent state of these cells. It often maintains them in what's called a quiescent state, an inactive state, until the tissue is damaged. And at that point, you have to have cues that allow these cells to become activated. At that point, they will start dividing. They usually regenerate a normal stem cell again. It's called self-renewal. But they will also generate progenitor cells that go on to differentiate into all different kinds of cell types that will exert the functions of our tissues, right? So in many cases, these are asymmetric divisions. You have one new stem cell and one differentiating cell. But there are also cases in which you have symmetric divisions when you, for example, need to grow the pool of stem cells that you need. We're actually currently investigating the decision-making processes that allow stem cells to divide symmetrically versus asymmetrically. And we found that indeed with age, these processes change. Unfortunately, I won't have time to talk about these aspects, but if you have questions about these aspects, you can ask me later. What I want to point out is that these cells are, again, they're sitting in most of our tissues. They are regulated at multiple levels. So they are, of course, what we call cell autonomous regulators, so signaling mechanisms, genes, pathways that control the quiescent state, they control the multipotent capacity of these cells. They will also control when cells can respond to a stimulus. And they will also control whether cells differentiate into one cell type or another. And these include mechanisms that control the epigenetic state of cells, or even things that control, for example, the redox state, the concentration of free reactive oxygen radicals in these cells that usually have signaling capacities as well. But there are also, as I mentioned already, niche factors from this microenvironment that will control stem cell activity and multipotency. And there are also systemic factors, factors that are in the circulation, growth factors, hormones that are in the circulation and will determine the activity state and the capacity of stem cells in multiple different tissues. And so you have to imagine that all of these different local and systemic processes have to be integrated by the system in order to achieve a response that is adequate to a particular situation. And of course, with age, all of these things also change. So there are intrinsic changes. For example, it's well known in vertebrate stem cell systems that you have an induction of cell cycle inhibitors that you can detect. There is, of course, telomere loss in stem cells that divide very often. So you end up with a situation where the ends of your chromosomes shorten over time, and you basically impact the capacity of stem cells to divide. That also leads to DNA damage. There are epigenetic changes. There are, of course, changes in the redox state as well. All of this will impact the functionality of these cells, sometimes in a positive, sometimes in a negative way. The same is true for the local support. There are changes that have been well described, actually, changes in niche-derived factors that usually maintain stem cells. They decline with age, and you end up losing stem cells in different tissues. And then, of course, there are systemic changes that we all know of. And these are quite exciting because we can imagine that maybe by tuning this extracellular environment a little bit, and I'll talk about this later in the talk when I'll discuss some work we've been doing with parabiosis, that maybe by tuning the extracellular environment, you can manipulate the capacity of stem cells to regenerate and maybe rejuvenate the system to a point where you are able to regenerate better than you normally would at a given age. And so we've been trying to work on these different aspects over the years. And I'll give you a bit of an overview of this, mostly focusing on what we've done in the intestine of the fruit fly. The intestine of the fruit fly is interesting because it has a population of stem cells that are relatively simple. They generate only a limited number of different cell types, so we can characterize it very well. And they're amenable to genetic perturbations that we can do very efficiently. And we can do this in a cell type specific and temporally controlled manner. So we can take an old animal and we can say, okay, this gene we will activate now in the stem cell specifically, and this other gene will inactivate specifically in the daughter cells. And we'll see what happens at the tissue level. So these type of manipulations, of course, are very difficult to do in vertebrates. Say if humans, it's impossible, right? So we have the fly as a model to ask very fundamental questions about how aging is impacting stem cell behavior. And then based on this work, we've identified a number of ways by which we can intervene to try and maintain stem cell function over time and maybe even extend lifespan. And then at the end, I'll talk about a study in which we've identified immune modulators, or one immune modulator that can improve regenerative capacity in damaged tissues. And it turns out to have very interesting longevity effects as well. And I'll give you some details there. So the fruit fly, of course, has an intestine. This is a fly that has been eating actually a pH indicator. It has been eating bromophenol blue, and then it was dissected. And so you can see that the gut is actually compartmentalized. You have the acidic region here, it's yellow, and an anterior midgut and a posterior midgut that are more basic, and therefore they're blue. All along the gastrointestinal tract, you have an epithelium that is, of course, important for food resorption, for nutrient resorption, but it's also important to maintain the microbiota and to interact with the extracellular environment and the outside world in terms of also interacting, of course, with viruses and with pathogenic bacteria. Therefore, this epithelium will have an important role in maintaining innate immune homeostasis, so it will actually respond with innate immune responses to pathogens. It's important to interact with good bacteria in the commensal microbiota. It's important to maintain permeability that allows nutrients to come in but not anything to come out. So it's a complex tissue that needs to be maintained properly. And these cells that are making up most of this tissue are basically enterocytes. You can see this in a cross-section of the intestinal epithelium. It's made up of a single layer of these enterocytes. This is one nucleus, and this is more or less one enterocyte. This is another enterocyte here. Large cells that really exert all these functions in this epithelium and therefore get damaged a lot, right? And accordingly, they need to be replaced, and they are replaced by stem cells that sit here between the visceral muscle, which surrounds the gut and allows the gut to move and peristalsis to occur, and the epithelium proper. So they sit here and are usually inactive, but will get activated in response to damage to the enterocytes, and they will regenerate these cells. A cartoon of this would look like this. So you have the visceral muscle, you have these enterocytes, and you have a second cell type here. It's seven differentiated cell type, which are the enteroendocrine cells. And they also are derivatives of these stem cells. So the stem cells will divide. They will give rise to a so-called progenitor cell. It's called an enteroblast. And this progenitor cell will differentiate into enterocytes or enteroendocrine cells. Now, I mentioned already that we have ways in the fly to perturb genetically these cells very precisely. We also have markers for these cells. So if we produce a micrograph here of a gut that was stained with specific antibodies, or in which we express a GFP, a green fluorescent protein in these cells, this is more or less how it looks like. You have stem cells and enteroblasts sitting as cell nests on the basal side of the epithelium. Then you have these large enterocytes here, and you have these enteroendocrine cells labeled in red here as well. So you can identify them, you can count them, you can look at their proliferative activity. So there's a number of ways in which we can characterize the behavior and the function of these cells. And because of this capacity to do this, we have learned a lot about how the intestinal epithelium in the fly regenerates. The stem cells themselves were identified first in 2006, so not even that long ago, by Micheli and Olstein. And what they found is that, yes, there are these stem cells. They get induced by damage. They proliferate and generate these enterocytes and enteroendocrine cells. And then over the last 10 years, many different labs have studied this extensively and characterized really what we call the lineage relationship between stem cells and the differentiated cells. So again, we know that they are mostly quiescent. They get activated by stress. They will end up dividing to generate a new stem cell and an enteroblast. And here, one of the recent findings is that the lineage of these splits very early, actually. Already in this active state, you have two flavors of stem cells. One that starts expressing this Prospero gene, and others that don't, but have very high levels of this gene called delta. And these delta high cells will generate enteroblasts that then go on to differentiate into enterocytes, while these prospero high cells will generate enteroblasts that go on to differentiate into enteroendocrine cells. So we understand the molecules that are driving the split in this lineage to generate two different differentiated cell types. More critically for us is that we can characterize their proliferative response to tissue damage quite well by, for example, exposing the animal to an enteropathogen. In this case, Ehrlichia carotovora 15, which is a mild enteropathogen. It damages the epithelium of the gut, but it will not kill the fly. The fly actually mounts an innate immune response, regenerates the epithelium, gets rid of the bacteria, and goes on living. And so we can look at how many stem cells in the epithelium are dividing at any given time point after the infection by just looking at the number of phosphohistone-positive cells. Phosphohistone is a marker for mitotic cells, and in the intestinal epithelium of the fly, only stem cells will become mitotic. And so we can, by counting the number of phosphohistone-positive cells, we know how many of these cells are active. For example, at 12 hours, we have a peak of activity where most of the stem cells will actually be dividing, and then over 24 hours, they return to a quiescent state. And this is a critical time course because it allows us to then ask questions about genetic pathways that control the activation step and genetic pathways that control the return to quiescence. Now, when we look at the aging animal, it's very clear that something goes wrong. This is a young animal in which, again, the stem cells were labeled with this GFP marker, which is labeling stem cells and enteroblasts. And we're looking at this, the basal side of the epithelium, so you see all these stem cells nicely dispersed and inactive at that point. In an older animal, 45 days is maybe a 50, 60-year-old human, accordingly. These animals will end up with a hyperproliferative state of these stem cells, which continuously divide. They will generate a lot of enteroblasts. They do not properly differentiate and end up piling up on the basal side of the epithelium, and you end up with what we call a dysplastic state. You can see this also in these electron micrographs. You can see these cells piling up here on the basal side of the epithelium. And this is really a phenotype that happens in every single animal over time. When it gets old enough, it will look like this. So clearly, it's something that is age-related and is driving stem cell misregulation. And we've tried to understand where this comes from. And over the years, we've learned that it's mostly a hyperactivation of an innate response to damage. In this case, a response that is driven by molecules that are commonly known as stress response molecules or inflammatory molecules, the JAK-STAT signaling pathway, the Jun kinase signaling pathway. These are pathways that are activated in stem cells or in their surrounding cells in response to tissue damage and will promote a proliferative response that you need in order to regenerate the tissue. But in an old animal, these pathways become hyperactive. And they become hyperactive because of excessive reactive oxygen species, excessive tissue damage, excessive inflammation, etc. So that drives this tissue into dysplasia. What we also learned over the years is that we can identify factors that allow us to limit that response. For example, we can overexpress a molecule called NRF2 or a molecule called FOXO that acts as a brake to the proliferative activity of these cells, dampens down their activity, and allow us to maintain homeostasis or regenerative capacity for a longer time, and that will ultimately lead to an increase in lifespan. One example is shown here, where we are overexpressing, not now these NRF2 or FOXO, but we're actually overexpressing target genes of NRF2 and FOXO. One is called JAFRAC1, it's a peroxyredoxin, an antioxidant enzyme. HSP68 is a chaperone, an enzyme that ensures that proteins are folded properly in the cell. If you overexpress these factors, you end up with the increase in proliferation that we see in the wild type with age is dampened. It's not completely abolished, but it is reduced. And this reduction is important to improve the health of the animal at older ages, and so you get lifespan extension in females and in males as well. And you can play this game with many different genetic perturbations, ultimately showing a correlation here between the proliferative activity of stem cells at a given age, in this case at 30 days of age, and the overall lifespan of the population. And this is now showing different genetic backgrounds. So this is where wild types would be. If we reduce proliferation rates of stem cells, we almost linearly increase lifespan, up to the point where you impair regeneration. And FOXO actually is so strong in preventing proliferation that it will actually inhibit regenerative capacity, and you end up shortening lifespan. The same is true for certain alleles of the insulin receptor. When you overexpress this molecule, which is actually a Jun kinase, it will activate the JNK pathway, and you end up with more proliferation and shorter lifespan. So clearly the proliferative activity of these stem cells is driving the dysplasia, and by perturbing the amount of proliferation, we can adjust the system towards homeostasis. And that was very important because it was actually the first demonstration in any organism that you could specifically perturb stem cells like this and have an effect on longevity overall. So since then, we've tried to understand in more detail where this dysplasia is in fact coming from. Because in one of the rules of aging research is you can always ask, well, why did this happen, and why does this happen before this? And so you can always go back until you're in embryogenesis, and then you don't know what to do anymore. But in this case, I think we have a relatively good understanding right now on how, with age, you develop an inflammatory condition in the gut that ultimately leads to this dysplasia. And what happens here is that so I've shown you before that you have this compartmentalization in the gut. The gastric region is acidic, and there are specific cells in the gastric region that secrete protons. They secrete acid in order to acidify the lumen of the gut. And it turns out that these cells are very sensitive to cytokines in the surrounding. And so with age, we know this also in humans and in vertebrate models, with age there is an elevation of inflammatory cytokines that one can measure. And it ends up activating a pathway called the JAK-STAT signaling pathway, which in these cells leads to what we call a transdifferentiation. So these cells, when they have high JAK-STAT signaling activity, end up transforming themselves into cells that are more common for the posterior midgut. And once you have that, they lose the ability to secrete protons. And that means that in the end, you end up with a gastric region that is less acidic than it should be. And that ultimately has an impact on the commensal microbiota. So the commensal bacteria that are in your gut are also in the fly's gut, let's put it this way, are actually also compartmentalized. There are some bacteria that are more common in the anterior midgut, some bacteria that are more common in the posterior midgut. Once you lose this acidic barrier, which they can't usually pass, you end up with mislocalization of bacteria in different regions of the gut. That ultimately then leads to a secondary inflammatory response in the posterior midgut that includes the activation of these players I mentioned earlier, the Jun kinase signaling pathway, the P38 kinase signaling pathway, the JAK-STAT pathway, now in the other region here. And as soon as you get this, you end up with tissue damage and excessive proliferation. And we can really try and characterize these mechanisms one by one and look at specific markers for change. So we can look at, of course, the transdifferentiation events. We can look at how the composition of the commensal microbiota changes. And then we can look at how stem cells are changing the proliferative behavior. And we've identified pathways that control these things. And the important thing here is we can actually perturb these pathways at different time points during this development and ending with lifespan extending interventions. So what this does is it gives us a relatively large or a clear or comprehensive view of the age-related decline in tissue homeostasis in a tissue like this. It has some parallels to human disease. Inflammatory bowel disease is a good example, where you have this interaction between the commensal microbiota and innate immune responses in the epithelium as well. And of course, it teaches us about how the local environment is influencing stem cell proliferation. All right, so I wanted to dig a bit deeper here on the question of stem cell maintenance and activity with age. And I want to talk about some work that Samantha Haller, a postdoc in the lab, published just recently. And it becomes a bit more complex, but bear with me here. In the end, the take-home message is very simple. What we had identified early on were signaling events that are important for the daughter cell of a stem cell to differentiate. And this signaling event is actually the TOR signaling pathway, activation of the TOR signaling pathway. I'm pretty sure you've heard about TOR signaling here before, in these 2 weeks, right? So TOR, of course, is a regulator of growth, and it actually promotes protein translation. The so-called enteroblasts, they need to grow. They grow about 100-fold or so when they differentiate into enterocytes. So they need high TOR activity. But TOR does not only allow them to grow, but it also actually promotes their specific differentiation into a different cell type. What this also means is that TOR signaling has to be kept low in the stem cells in order to prevent TOR from driving them into differentiation. And that's, of course, a bit of a challenge because TOR is a very important molecule that controls proliferation, it controls growth, it controls translation, it controls nutrient responses. So you have to have a very tight control on this molecule. In the stem cells, we learned that they express high levels of a negative regulator of this TOR molecule, TSC2, and that actually in the enteroblasts, the same TSC2 gene is specifically downregulated in response to differentiation pathways. Okay, so that was published in 2012. We had a relatively clear understanding for how you had to suppress TOR in stem cells and then to prevent them from differentiating, and then you needed to activate TOR here. Now, the surprising thing, though, was that in the same study, we had asked whether TOR deficient stem cells would behave somewhat differently than normal stem cells. And the way we do this here is we basically induce a mutation in the stem cell and then ask, through a so-called lineage tracing approach, we ask how many cells does this specific stem cell generate in the course of about 7 days. And if you do this, you can basically count the number of cells that every individual stem cell has generated over these 7 days. This is what our wild-type controls look like. A normal stem cell generates between 10 and 15 cells in these 7 days. An insulin receptor mutant stem cell does not. It actually just sits there and doesn't do anything. So you have single-cell clones. But TOR-deficient stem cells also don't generate a lot of daughter cells, suggesting that in the stem cell, where we know TOR has to be suppressed, right, that TOR has a role there in maintaining normal proliferation. And that was somehow contradictory. So we decided to look at this a little bit more carefully. And we used this bacterial challenge model that I mentioned earlier to ask, well, how fast do stem cells actually respond to a challenge in different conditions? In this case, in a condition in which we eliminate the TOR signaling pathway. This is one molecule that is important for the activation of TOR. If we eliminate this molecule, what you can see is that the normal response here in black to the bacteria is significantly shifted to a later time point. So you need more time for the stem cells to respond to this challenge than when they are in a wild-type condition. At the same time, we can detect TOR activity early in this regenerative response. This is in wild-type stem cells. This is a marker for TOR activity, phosphorylation of the 4-ABP molecule. So you can see this light up here, while in the non-challenged animals, there's nothing there. So we have a transient activation of TOR early in the regenerative response that seems to be important for the rapid proliferation that occurs after a challenge. And that, to us, looked very similar to a paper that was published by Tom Rando, who ended up being a collaborator of ours, in which he had looked at, or his lab had looked at, the role of TOR in muscle stem cells in mice, satellite cells in mice. And what they had shown is that, so mTORC1 is the same as TOR in flies here, that this molecule is required, that the TOR pathway is required to allow a satellite cell to exit quiescence, to be activated rapidly, because it promotes a transition into what they call the G alert state, a state in which they are primed to divide. They're basically ready to respond. And so the actual data that they had in this paper was quite similar in terms of the proliferative response as we had seen in the fly intestinal stem cells. Now, we asked a different question, though. Again, we knew that if you activate TOR in stem cells, you can drive them into differentiation, which means you lose them. So we asked, well, if that's true, then every time you activate stem cells, you're going to run the risk of losing them to differentiation, because now you're activating TOR every time. And so we asked whether we could actually test this. And in fact, we can, because we can, as I mentioned, the flies don't die from this ECC15 infection. So we can ask whether repeated rounds of infection, where you force the stem cells to divide, whether this would end up with a significant loss of stem cell numbers. So we're not counting the number of stem cells here. Even after 2 infections compared to the control, we already have a significant decline. After 3 infections, it gets worse. After 4 infections, it gets even worse. And we knew that this is TOR dependent because we can inhibit the TOR pathway by overexpressing negative regulators. And now you actually get more stem cells after 3 infections, because now you're actually forcing symmetric divisions. Now the daughter cells cannot differentiate, so you get more stem cells. If you activate the TOR pathway by knocking down this molecule, you end up losing the stem cells even without the infection, right? Okay, so the role of TOR in promoting this proliferative response can also be deleterious in the context of the numbers of stem cells that you have per tissue. But you could also think of this as soon as you start using your stem cells, you lose them, right? So you don't want to force them to regenerate too much. And we wanted to know whether, in fact, because it was so similar to the satellite cell study, we wanted to know whether, in fact, we can ask the same question in mammalian tissues. And for that, we went into the airways. We actually looked at the tracheal epithelium of the mouse, mostly because the tracheal epithelium looks very similar to the fly intestine. You have stem cells that sit on the basal side of the epithelium. You have only a limited number of differentiated cells. And actually, this is a cartoon that just highlights these similarities. But more importantly, the lineage relationship between the stem cells, their activated conditions, their asymmetric division, the differentiation of the daughter cells into differentiated cell types, all of that is very similar in the mouse airway as well. Even the role of these molecules, so delta and notch, in driving differentiation was conserved. So it was really for us a test case to say, okay, can we ask whether similar stem cell types in vertebrates are affected in the same way as the fly's intestinal stem cells are? And in fact, if you damage the epithelium of the mouse airway, and you can do this by exposing the animal to SO2 gas, you end up seeing a similar TOR response. What happens there is that you kill all the differentiated cells of the epithelium, but the stem cells will stay as a single layer of cells. So this is the control tissue. These small white cells here are the stem cells, and then there are these other differentiated cells that make up the rest of the epithelium. One day after an injury with SO2, you can see it's only one layer of cells. Now these cells are pretty large. They still have stem cell markers, but they also light up when you stain them for phospho-S6, which is a marker for TOR activity. So you end up with high TOR activity here. And they all become proliferative because this green marker here tells us that they are proliferating. There are very few proliferating cells in the control epithelium. After one day, they're all proliferating. And then after 3 days, it's hard to see here, but you have a reestablishment of the normal epithelium with stem cells down here at the basal side, very few proliferating cells. So you have, again, a transient response that we can monitor. And it goes along with an activation of TOR in the stem cell population. So we asked what happens if you now drive TOR activity constitutively for a while. And we can do this genetically in the mouse now by eliminating this negative regulator again, TSC1, through a keratin 5 cree. Keratin 5 is a promoter that's expressed in the stem cells, in the basal cells. So we're knocking out TSC1 now specifically in the basal cells, which leads to their activation of phospholipase 6. So you can see high levels of phospholipase 6 in these cells. You can count the number of phospholipase 6 positive cells as well. But you can also see that these cells now become large and they become proliferatively active. So this is the number of Ph.D. positive cells in these animals. Now all of this was analyzed at 10 days after the induction of TOR activity, after the knockout of TSC1. But if you look at this later, at 28 days, the one striking thing you see is that most of your basal cells, which are here labeled in black, are gone. And this is quantified here, so you can count this. And they're gone, we think, because you have constitutively activated TOR and you're driving them into differentiation. So of course, we can also then ask the same question we did in the fly. If we repeatedly induce an injury and we force TOR activation, do we see a loss of basal cells? And indeed, that's the case. So this is a comparison between animals in which we expose them once to SO2 versus exposure of 3 times to SO2. You can see that the number of basal cells declines significantly, but not if we give these animals rapamycin-containing food. And rapamycin is a drug that can inhibit the TOR signaling pathway. And so you can see that there we maintain basal cell numbers quite nicely. So I was talking about this in the context of aging. So far, these are not old mice, right? But actually, if you look at old mice, you can see that one of the striking features of the airway epithelia, and this is actually a low magnification image, so this is actually the whole airway here. What you see in white is the stem cells labeled with this marker. In 24-month-old animals, you see very few of these still labeled. This is some background staining here. This is a high magnification. So in green are the basal cells. In the old epithelium, there are very few left. And this is just wild-type aging mice. Well, what happens if we expose them to rapamycin? Well, in fact, we can do this experiment at any given time point. We can start them at 9 months of age and then harvest the tissue at 12 months. We can start them at 12 months and harvest it at 24, or we can start them at 15 and harvest them at 18. In any one of these conditions, we end up rescuing the number of basal cells that we see as a loss, normally, in the old animal. So the model here is quite simple. In a normal young tissue, stem cells need to activate TOR signaling in order to respond appropriately to a damage signal. And TOR activation is important for this rapid response. But at the same time, because of TOR's role in driving differentiation, that activation of TOR has a deleterious consequence, which is that stochastically you start losing many of your stem cells. Accordingly, in old animals, where you either have repeatedly gone through regeneration or in some cases you even get chronic TOR activation, you end up with a loss of stem cells that you can rescue then if you limit TOR activity, for example, by rapamycin exposure. And of course, you may have heard this in the course that rapamycin is one of the few drugs that is clearly extending lifespan in a wide range of different animals. All right, so, okay, I have 20 minutes, right? Well, 15. 5? Something like this. I'll go through quickly here. So in the third part, what I wanted to talk about is, as I mentioned, an immune modulatory process that allows us to improve regeneration. Initially, we found this in the retina, but we've expanded this work now, and I'll give you some highlights of that work. So the concepts here are a little bit different. We were interested, I mean, my lab was interested for a long time in trying to understand how light can damage photoreceptors and how photoreceptors are actually deciding whether they should go into apoptosis or not. And so we've been doing fly genetic screens to characterize apoptotic pathways in these. And we found a gene that was interesting. I'll talk about this in a second. But a colleague of mine at the back was actually asking a different question. He was trying to develop methods for cell replacement therapies in blind mice first, but potentially in blind patients, of course. And the way this is done is you basically generate photoreceptor progenitors from induced pluripotent stem cells or from embryonic stem cells. You inject them in the subretinal space, and then you hope that they integrate into the retina. And I say hope because most of the time they don't. And they don't do this, especially in the conditions in which you would like them to do it, which is in the diseased or aging condition. In a young animal that is not diseased yet, you end up having relatively good integration efficiency. You can see these photoreceptors go into the retinal photoreceptor layer and have functional connections as well. But in an old or degenerating retina, this integration efficiency is significantly reduced. And so, because of the work we're doing on trying to understand photoreceptor survival in flies, and where we had identified molecules that can change the immune environment and therefore promote regeneration, we asked whether these same molecules may be able to influence the immune environment to a point where you inhibit inflammation, and thereby you might be able to increase the efficacy of cell replacement therapies. I'm going to skip this. This is just to introduce the fact that the retina can actually attract immune cells, and that there are two different types of immune cells, a pro-inflammatory immune cell and an anti-inflammatory immune cell. And that, of course, the strategy here is to try and reverse this process. With age or in a degenerating retina, you get a lot of these pro-inflammatory cells, and you want to reduce that activity. So what have we shown in flies? In flies, we had found in a simple assay in which we basically irradiate one side of the developing animal with UV light, and then we ask how much tissue has been lost once the animal develops as compared to the control eye. So this is a screen now. We can actually genetically perturb this eye and ask, well, in relation to the other eye, do you lose less tissue? So is regeneration improved? Or do you lose more tissue? Is regeneration reduced? And so in these type of screens, we found that, in fact, if we have flies that have no hemocytes, hemocytes are immune cells in flies. They're in green here. So they're dispersed throughout the animal. And these hemocytes actually get recruited to the damaged eye and improve repair. If we kill these hemocytes, or hemoless flies, we'll therefore end up with a significant increase in tissue loss. So hemocytes were important, and what we had found is that the damaged retina actually recruits these hemocytes and promotes their transformation into what we could call an anti-inflammatory phenotype. And this is done by a molecule called MANF, mesencephalic astrocyte-derived neurotrophic factor, which actually gets transcriptionally induced in these hemocytes in response to the damage, promotes this transformation in an autocrine way. So it's actually secreted from the hemocytes, feeds back on them, and promotes the transformation into an anti-inflammatory state. And you can view this as a negative feedback loop. So you're attracting all these pro-inflammatory immune cells. They start secreting this factor in order to shut themselves down, because after you have elicited the initial inflammatory response, you want to actually have a break on it in order to promote repair. Right? And that's what MANF is doing. So in the fly then, if we actually eliminate MANF from hemocytes, you lose more of the eye. If you overexpress MANF in hemocytes, you get more, you rescue the phenotype. And we could show, as I mentioned, that this factor can actually promote the transformation of these cells. For example, when we take these cells in green here and put them in culture, after the UV treatment, you see these large red cells. These are the anti-inflammatory-like hemocytes. If we overexpress MANF, you get, this is without the UV treatment, you get the same effect. If you actually take these fly cells and put human protein, human recombinant MANF into the culture, you get the same phenotype. In the mouse then, we asked, well, what happens if you inject MANF into the retina? And well, if you do this, you start attracting a lot of immune cells to the retina, and they start to express markers for anti-inflammatory polarization. So you can also do this in culture. You can see activation of these markers after MANF treatment. More critically, if you injected MANF into the retina, and then you damaged the retina of the mouse by high light exposure, in this case you're using bulb C mice, which are very sensitive to this white light, In normal conditions, you see a lot of apoptotic cells, tunnel-positive photoreceptors, cells that have decided to undergo programmed cell death. But if you inject MUNF, you can block this apoptotic response. And we can also, in the paper then we showed that this is also then improving the maintenance of the retina in other degenerative conditions as well. But we really wanted to ask whether we're able to improve the efficacy of these cell replacement therapies. I mentioned that you can see these green cells now integrating into the photoreceptor layer in a mutant animal that shows progressive photoreceptor decline. You can inject the green cells in the subretinal space, but you don't get any cells into the photoreceptor layer. But if we co-inject MUNF, now you can see the cells coming in. This is just green cells, but you can actually ask whether you have a functional consequence of this by, for example, doing an electroretinogram. We basically measure electric activity in the retina in response to a light stimulus. And indeed, when you actually, do it this way, it's easier. When you actually look at a blind mouse, of course, there's no response. If you inject photoreceptors without MUNF, the response is minimal. But if you co-inject MUNF, over time, you see a restoration of visual function. Suggesting, in fact, that the therapy here, which is the injection of photoreceptor progenitors, is effective in this context. So what this suggested is that, yes, we can actually use invertebrate model systems to find therapeutically promising molecules that can allow us to improve replacement therapies in this context. Now, MUNF was actually discovered a while ago, but nobody really understands clearly how it works. There's some suggestion that it can respond to ER stress. It has anti-apoptotic activity in culture, specifically on dopaminergic neurons. But mostly the receptors and signaling pathways are unknown, and so that's something that we're doing right now. But one thing that we noticed, actually, is that with age, MUNF levels decline in mice and in flies. This is whole fly protein extract, and you can see that at 50 days of age, there's much less MUNF in these animals. In the mouse, we have also protein extract, skin, adipose tissue, liver, and muscle, much less MUNF than in the controls. More importantly, circulating MUNF in the plasma, this is using an ELISA here, declines with age significantly. And if we take human serum and we look at the levels of MUNF, you can see that with age, with increasing age, there's also less MUNF in your circulation. That's, of course, a correlation. So we wanted to know whether the declining levels of MUNF systemically would have an influence on longevity and on tissue homeostasis. In fact, if we knock down MUNF in the fly, we end up shortening lifespan. These are these red lines here. We shorten lifespan in conditions even, or what happens here is actually you get increased dysplasia. This is the proliferation activity of stem cells. And it's actually inflammatory signaling pathways in the gut. You can see these upregulated. And here you see the dysplasia phenotypes. More importantly, I don't want to go through all these data, but more importantly, when we overexpress MUNF in adipose tissue, in hemocytes, or in cells that activate expression of MUNF when they are stressed, so we're using a stress-inducible GAL4 here, you end up extending lifespan quite nicely. But in neurons, when we overexpress MUNF, we see a shortening of lifespan, which we don't understand yet. So there's some tissue specificity here as well. Critically, when we actually look at mice that are deficient in MUNF, so in this case, we're using MUNF heterozygotes that have about 50% of the normal MUNF levels, they show an infiltration of immune cells. This is in the white adipose tissue, the liver, the pancreas. All these dots here, these are all immune cells. Many of them are CD68 positive, which is a marker of pro-inflammatory conditions. And if you look at the liver, what you see is a massive increase in lipid vesicles. So you basically have a fatty liver disease in these animals. You have liver steatosis that develops within 10 months of age. Now, if we do the reverse, if we overexpress MUNF, in this case, we overexpress it in the liver specifically, and then we feed the animal a high-fat diet, which leads to liver disease anyway, you can see that you can restore or we can reduce the amount of fatty liver disease in these animals. Furthermore, if you actually knock down MANF or you reduce MANF levels, and then you look at liver damage markers, phospho-JNK is one marker. It's increased in animals that have reduced MANF levels. The number of tunnel-positive apoptotic hepatocytes is increased as well, suggesting that in an animal that is deficient in this molecule, you end up developing a damage phenotype, an inflammatory and damage phenotype in the liver that is actually seen also in old mice. And so we, I'll skip this. We started looking more broadly at markers for aging. In this case, we're looking at senescent cells in adipose tissue. In the MANF heterozygotes, the blue color here suggests that there are more senescent cells. You can also see an increase in circulating inflammatory cytokines. IL-6 and IL-1 beta are increased. Oh, actually, this is from white adipose tissue, but you can also see this in circulation. And so what this suggested to us is that deficiency in MANF leads to very broad inflammatory effects that you also see in an old animal. And so we asked whether this means that MANF in circulation plays an important role also in the rejuvenating effects of parabiosis. So when you join the circulatory environment of a young and an old mouse, the old mouse receives some factors that are not understood from the young mouse that rejuvenate some of their tissues. The reverse is also true. Some of the young tissues get older as well. So we asked whether if we do this in a condition in which the young parabound is MANF heterozygous, do we still see these rescue effects? Well, this is actually looking at liver fibrosis here. You can see that in the normal heterochronic parabosis, you rejuvenate the liver, but in the MANF heterozygous parabound, you don't. And you can measure this with multiple markers. For example, phospho-JNK levels are rejuvenated, but not with MANF heterozygous. Tunnel-positive hepatocytes go down normally, but not with MANF heterozygous. CD68 positive cells in the liver go down, but not with a MANF heterozygous. The same is true for gene expression. I'm not going to bore you with this. And then finally, we did one experiment where we injected MANF into the intraperitoneally into the mouse and asked whether we could rejuvenate some of the liver phenotypes in the old condition. And in fact, all of these markers also are rejuvenated. Sorry, this is MANF overexpression, and this was the IP experiment, but it's ultimately the same result. So what we're seeing here is a factor that seems to be systemically regulating what we call immune homeostasis. It declines with age in multiple animals, and we think contributes to aging phenotypes, probably not just in the liver, but we focus on the liver for now. We need to analyze other tissues as well. And that by restoring normal MANF levels in circulation, we're able to rejuvenate some of the phenotypes, at least, that we see in the aging animal. All right, and with this, I want to end and just thank people that did the work. Joanna Neves did most of the work on MANF, and Samantha did most of the work on taurin stem cells. I didn't really have time to talk about anybody else's work. Thanks. Yes.
I have three questions. Do you have macrophages in the gut in the ananormal conditions, or do they come as M1 and M2?
You're talking about the fly gut or the mammalian? In the fly gut, we have actually done some work where we see that these hemocytes, which are basically macrophages, they are recruited to the gut in response to both bacterial damage, but even oxidative damage. So tissue damage in general recruits these hemocytes. The hemocytes interact with the stem cells by activating the proliferation, actually, and then they leave the gut again. We know what the signaling pathways are that control this, but we haven't studied further how tightly they associate with the stem cells or with other tissues and so on. It's also not so clear what the role of these hemocytes is in, let's say, fighting bacteria specifically. So there's still some work to be done. But there's clearly a transient association there.
One question about energy. The factors you had for regulating the stem cells, there was oxidative stress, but metabolism?
Yeah, it's this person is doing this right now. So she's looking specifically at metabolic readouts in stem cells, and she's looking at whether metabolites from the commensal microbiota may be influencing regenerative activity, which in the fly we can do relatively well, but it is a fairly complex question to address, right? Because you're looking at, we still don't understand the metabolic activities in the microbiota, for example. We have to identify these first, then we have to ask which of these metabolites are actually essential for regeneration of stem cells, all this kind of stuff. So we're doing this right now, but I expect it to be take some time. What we have done on top of this, though, we've looked at the role of calcium in regulating stem cell activity. We had a paper a while ago showing that stem cells actually show oscillating levels of calcium in the cytosol, and that in response to activation to mitogenic signals, calcium is released into the cytosol, activates CRTC, and you end up requiring this activation in order for proliferation to occur. Now, calcium, of course, is stored in the ER, but it's also stored in the mitochondria. And we have, so this guy Otto here, he's actually exploring the role of mitochondrial calcium uptake in proliferative activity of stem cells. And indeed, you actually require what is called the mitochondrial calcium uniporter for the proliferative response. And what we're doing right now is we're trying to understand what the elevation of calcium in the mitochondria, what consequences that has on mitochondrial metabolic output, because I think that the metabolic output of mitochondria needs to be adjusted, of course, to the proliferation state of the stem cell, and that calcium may play a signaling role there to actually control that balance. And we have some initial data suggesting that indeed NADH levels that are emerging from mitochondria, or NADH levels in mitochondria actually are different depending on which calcium concentration you have. But it's, that also is going to take a little bit.
One last question. Is it the regulation of your stem cells, where do the factors come from which suppress the mTOR pathway? Do you have a niche or is this autocrine?
The TSC complex, of course, is intracellular, right? So we don't understand the regulation of TSC in this complex, in this context very well. We know from other studies, mostly in development and growth, that there are a number of extracellular regulators that will activate the TOR pathway, amino acids, for example, and so on. But what we don't know is whether the inhibition of TOR is an active condition or is basically the default, which may well be. All we have shown is that in the differentiating cells, TSC is actually selectively repressed in order to activate TOR. So it's entirely possible that the system is set up so that TOR repression is the default state, and that you need all these inputs in order to actually activate it specifically. At least we don't know of any active regulator that will suppress its activity from, I mean, I don't know if you know any of it. So. Yeah.
Okay. Are there any human therapies that you can envision eventually growing out of this? I mean, mAb pills or TOR pills or whatever?
Well, there's a reason they hired me at Genentech, right? So that's basically what they want us to do. We are trying to learn more about the regulation of MANF and what the receptors of MANF are. MANF as a therapy itself, at least in my current job, I couldn't do because there's another company that has the patent on MANF, which is fine. But because I think what one really needs to know is what are the signaling pathways that mediate the effects of MANF. And then you have a whole series of potential targets that you could hit there, right? So that's really, it's definitely one thing we're thinking about, because the effects of MANF seem to be very moderate. So if you actually compare so I'm talking about MANF as a it's a factor that induces an anti-inflammatory state in these macrophages, which is basically a polarization into a M2-like state. Classically, there are specific interleukins, IL-4, for example, that will induce that state. If we compare the response of macrophages to IL-4 and to MANF, it's like night and day. I mean, IL-4 induces massive amounts of markers and cytokines that are M2-like. MANF does this to a level of maybe 1 to 5% of what IL-4 does. Now, the problem is IL-4, if you inject it into an animal, kills the animal. MANF doesn't. So maybe it's this tweaking of the response that we really need to understand. This very moderate modulation of the anti-inflammatory state. And so that's what we're trying to explore right now. But, you know, it's early times. Yes?
You said that the man blood was increasing with it. Do you know why?
No, we don't. We don't at all. We don't even have a good theory for why they're declining. See, that's the problem with aging research, right? Because you can always ask, well, why is that? For MANF, no, we have no idea. The biggest problem is that MANF is expressed in many different tissues. So even if we see less in circulation, we wouldn't know where it comes from. It also gets induced by damage. So one hypothesis could be that it's not actually steady state levels that decline with age, but it's actually the ability to induce it that is reduced with age. But we have no idea yet. So it's certainly something we need to start exploring. I mean, the fact that MANF declines with age is something that we're just now trying to publish, and then we'll probably go further. So. Yes?
What have we learned about telomere size and aging? I mean, some 10 years ago, it was like,
Yeah.
thought there was a real connection.
Mm-hmm.
What have we learned across species?
No, there is certainly a connection. I mean, there is beautiful work actually from Leonard Rudolph, which you know, who has looked at the role of telomeres in stem cells in the mouse intestine. And clearly, if you take, and so mice have very long telomeres, and you have to, when you make them deficient for telomerase, you have to go through multiple generations until their telomeres are short enough to get an effect. But once you have that, what you see mostly is ultimately a DNA damage response, right? So now the chromosome ends do basically fuse or induce a DNA damage response that will then lead to a standard either cell cycle arrest or misdifferentiation of stem cells and so on. So you can get all that experimentally. Now, is there evidence that telomere expansion can extend lifespan in mice? I don't think there is. I don't think anybody has.
Bad idea.
Yeah. Well, the biggest problem, of course, is you can induce cancer with this, right? So, I was at a meeting, on a longevity meeting, where there was a person called Elizabeth Parrish, who is well known apparently in the rejuvenation community because she claims to be patient zero for the longevity therapies. And so what she did is, as a private company, she went down to Colombia and injected herself with adenoviruses that carry telomerase and will extend her telomeres. And she measured it, they did extend her telomeres. I would not suggest anybody doing that because ultimately we don't know what the consequences are. And the best, I think, outcome for her is probably that maybe the effect is transient and she wouldn't get much of a deleterious effect. But ultimately, if you play with this, you end up really increasing the probability that your cells will become cancerous. And so, especially when you do this in stem cells, you might end up with this condition. So, all of this to say, basically, we need much more work to try and understand the intricacies of regulation there, and to understand how cells that are requiring telomeres, like stem cells, how they are dealing with shortening over time, how they respond to it, and how we can ameliorate these responses in order to maintain regenerative capacity. So just increasing telomere length is probably a bit of a hammer on something that one could do probably much more precisely, if we understood it better.
Folks, Heinrich put some of his neurotropic some of the wine out.
All right, let's go for it.
Thank you.